Hello everyone, my name is Carrington Vickers. I am a senior here at Hiram College, and today we will be looking at will carrots cause apoptosis in HL60 cells. This was done in molecular and cellular biology lab in fall of 2018 with Professor Maxim. So a little introduction before we get started into my results. So apoptosis is the death of cells that occurs as a normal and controlled part of an organism's growth or development. So this would show signs of blebbing, shrinkage of cell, and fragmentation. So to the right, you guys can see this photo, and it has the normal cell, what it should look like, and it has different stages of apoptosis that a cell could go through. And if you see at the end, it says necrosis, and that is bad. You don't want that in your cell because it's a cell death that is triggered by an external factor or disease such as trauma. We looked at human leukemia cells, which are also known as HL60 cells. These are promyeloid progenitor cells that normally differentiate into myeloid cells, a type of white blood cell. But in leukemia, it gets stuck in the undifferentiated state to divide uncontrollably, causing acute myeloid leukemia. This happens in bone marrow most of the time. And acute myeloid leukemia is a type of cancer, which is a genetic disease involving mutations in tumor-promoting and tumor-suppressor genes. And normally, radiation and chemotherapy cause significant damage to DNA, leading it to the death of the cell. But here we are looking at different ways to cause death to the cell so that people don't have to go through radiation or chemotherapy. They can do a more natural treatment plan. An important part of this project was WST8, which we treated our cells with this because when it's added to cells, it will produce a water-soluble yellow/orange colored dye upon its reduction in the cell. And visible cells will be seen at absorbance of 450 nanometers. So we looked under the microscope for this. Another important item I want to introduce before we get started is COMETs and COMET assays. So COMET assays help visualize DNA damage in the individual cells. So healthy DNA would just be the comet head, and damaged DNA forms a comet tail. So we will look at that in my results also. I just wanted to bring that up before we get to it, so you had a little background. And why did I choose carrots? So I looked at a paper called Bioactive Chemicals from Carrot Juice Extracts for the Treatment of Leukemia. And here they looked at carrot juice extracts where they treated myeloid cells for up to 72 hours. This induced apoptosis and inhibited progression throughout the cell cycle. So I wanted to look at this in my study, but instead I used raw carrots that were cut up, and I got the extract from them. So looking at my t-test, this data was generated using the WST8 reduction to determine cell viability and measurement at 450 nanometers. These cells were treated for 24 hours in this figure, and in the graph, these are the average absorbance values in treatment, and the t-test only showed significance when using the Campathin, which was our positive control. So next, let's take a closer look at the cells. So on the left, These cells are after 24 hours of incubation in the cyto16 dye. This dye is a fluorescent DNA binding dye which allows you to visualize nuclear condensation and blebbing. So here you can see that we did see forms of apoptosis, and there could be more apoptosis in the neon cells, but in the brightness we had on the microscope, they're not able to be seen to see if there's apoptosis, because if we were to dim down the brightness, then we would have lost all the other cells that I just circled. When taking a look at the cells after 48 hours in the dye, we see more apoptosis, but we also see mitosis. So I don't know exactly what this means, but it would be interesting to look at even further. So moving on, I wanted to show you guys the 70% ethanol versus my carrot extract, because in the ethanol you can see all these, what the healthy cells look like. Yes, there is a few that have maybe apoptosis happening where they're shrinking up, but not nearly as many as my cells had in the carrot extract. Next, we looked at comments under the microscope, and based on the chart, one, I did not have enough comments to rate because it said you needed 50 or more about to be able to rate them. But based on my 3, I don't think I have a comment, so I would give it a 0 because it should have like a tail right here. As you can see in some of these, like they're fragmenting off, and that's what creates that tail. It's the damaged DNA. So it looks like mine is pretty healthy. So in conclusion, I would probably determine this to be good treatment, but I would also like to take way more samples and look at it more closely. We only had about 3 to 4 weeks to work on this project, so we did not have a lot of time to do different things with it. So for future direction, I would like to have more time to take more samples. This way I can test it at 12 hours, I can test it at 24, 48, 72, and just keep looking at this, hopefully the same cell plate and cell vision to see what is going on with these cells when we do give them carrot extract. Also, I would like individual well plates because we used the well plates as a class. People might have been able to put their treatment into my well on accident, causing me to have greater significance and greater apoptosis than maybe what my actual extract would have been. Also, this just helps with contamination so that the moving back and forth from the incubator to the hood might cause something to go wrong with my cells. And also, lastly, I would like to try carrot juice because that's what the actual study looked at, so it's going to be interesting to see my data versus the carrot juice data if I was to do this again in the future. So here are my references. Hopefully you guys enjoyed this presentation, and one day you guys might look at if carrot extracts cause apoptosis in HL60 cells.